And thank you, Professor Reineke, for the very engaging talk. Therapeutic formulations and delivery is indeed one of the very apex problems of biomedical research, and your talk today has given a very insightful overview into non-viral nucleic acid delivery platforms. We have a few questions pouring in, and before proceeding with that, I would like to take this opportunity to thank Dr. Sriranjani Pulakat, my co-moderator, who has been compiling these questions for us. And now, without further ado, let me jump right into the first one. Are there any disadvantages to polymer gene delivery formulations over viral formulations?
Yeah, that's a really good question. So right now, I would say the disadvantage is that, there's been a lot more clinical movement with viruses, and there's a lot more clinical FDA approvals of viruses. And a lot of it has to do with the fact that it's been studied for a lot longer. And it's taken more research to really understand how to make polymers as effective as viruses for infecting cells. So as you all know, and we all know, unfortunately, viruses have had millions of years to perfect their genomes to get their infective DNA into us. And we have taken advantage of that for, really, modulating these systems. Whereas we've had to do a lot of work fundamentally to understand how polymers infect cells in a benign manner, such that, they're not toxic to us, but they're just relaying the good payload. So there's still a lot of work to do. There are some examples in clinical trials, like various modified variants of PEI or polyethylenamine. But I think the main limitation is just one, understanding specificity and targeting, and, just getting investors to invest in companies in this space, in kind of a tough biotech market right now.
Okay, thank you for that. The next question is, can these strategies be employed for delivering cationic compounds other than nucleic acids in a selective manner to avoid side effects?
I'm sorry, can you repeat the question? It kind of cut out a little bit on my side.
Sure. Can these strategies be employed for delivering cationic compounds other than nucleic acids in a selective manner to avoid side effects?
Oh, that's a good question. So generally the payload is anionic, so there's a non-covalent interaction. But I guess if I'm understanding the question correctly, so one is, yes, it could be used to deliver other anionic, various anionic species, even small molecules. So my group has collaborated with Aaron Carlson's group, where they're just looking at single nucleotide probes, and we've been delivering those. However, with respect to modulation of the backbone chemistry, such that if you want to deliver something else that's cationic, you could modulate that backbone chemistry, deliver the polymer into the cell, and make a cleavable linker off, such that you're releasing the cationic species. So I think there is ways to modulate chemistry and be very creative, such to deliver various ionic payloads into cells in a creative manner.
Okay. So moving on to the next one. How about delivering through the blood-brain barrier? How effective are these polymeric systems?
That is a really key question. I think that's one of the holy grails right now of the biotech industry, is trying to figure out how to pass big things through the blood-brain barrier. So right now, my group has not focused on that. There's been some groups like Susie Pun's group at the University of Washington who have done work and understanding sort of neuronal delivery and ways to get things through the blood-brain barrier. As of right now, it's tough to get something so large through the BBB. People are looking at alternative methods of administration, whether it's direct intrathecal or some central nervous system direct injections, intranasal, inhalations. or direct brain injections, I think is probably what's really state of the art in this. And it's even difficult to get viruses, so it's not just limited to polymers or liposomes. However, I think there are researchers looking at ways to try to pass this and modulate active transport across the BBB, but it's a tough barrier. It's designed to be tough because it's designed to protect us. But I think right now, direct injection into the affected area is really what state of the art is.
Okay. Next question is, are cellular uptake pathways of the polyplexes also influenced by their payload?
That's a good question. That we are starting to study. Okay. To our current work, we have seen that generally the uptake pathway is modulated by the polymer or the encapsulating species. However, I will say we have not done extensive modulation of the payload. But to the best of our knowledge, as of right now, and this obviously could change as we do more studies, we are seeing it's more modulated by the encapsulating package or the packaging, delivery vehicle.
A related question to that is, how do polyplexes fare in terms of loading capacity and transfection efficacy when compared to liposomes, micelles, or lipid nanoparticle-based delivery systems? And what are the major factors affecting the nucleic acid loading capacity in the lipid nanoparticles or polyplexes?
That's a very good question. Yes, so I do think that polymers and liposomes have similar loading capacity. So generally this depends completely on the size of the payload. So for example, you could take a humongous plasmid and make a polyplex or a lipoplex or LPN, lipid nanoparticle, and you would generally encapsulate one plasmid per particle. You could do this with siRNA, which are very short, this is millions of molecular weight. siRNA is maybe about, 19 nucleotides. That could contain hundreds of siRNA. So it's really kind of the package generally and loading capacity is dictated by the size of the payload. So there's also ways to formulate. So generally with lipids and polymers, we're drawing upon, decades and decades of work in the fundamental colloid formulation field, where people have really looked at formulation and colloids for a large number of years. And so when you formulate species, if you formulate them in really dilute conditions, you can get smaller colloids. If you put in ionic strength modulators, so like salt or serum, depending on the chemistry, you can aggregate these or grow bigger colloids. If you formulate at higher concentrations, again, the colloids can be bigger. So concentration, ionic strength, any other solution parameters, you can kind of modulate this, and you have to make sure you're essentially doing this for the exact chemistries you're looking at, because it can change. There are some rules, but generally the rules are variable based on the chemistry of the payload and the package. So what is kind of convention right now is that loading capacity can be varied. It can actually get quite high, but it is modulated by the size of the nucleic acid. But generally people are characterizing this with things like static light scattering and doing Zim plots, and also things like electron or cryo-TEM, where you can actually physically see and the payload numbers. So for example, with this CRISPR-Cas9 work, we did some work where we had a package of like a polymer or micelle. We loaded in Cas9 proteins, and you could physically see the proteins like encapsulated, and you can actually physically count them on a cryo-TEM image. And then we can do like Zim plots and molecular weight. So it's suggested right now to like not ever assume what the loading capacity is, but it is quite variable, which is a high advantage to this field, but it's always recommended to characterize these with different techniques.
Wow, thank you for that. The cryo-chem analysis sounds very fascinating. Yeah, a very related question again on this was the polyplex size considered as a factor in the machine learning based optimizations? And can the same machine learning or computational strategies be extended to other nucleic acid delivery platforms like liposomes or lipid nanoparticles?
Yes. So in short, yes, we did use size in our ML. You know, interestingly, a lot of our ML was based on cell culture. So in fact, in the culture, larger size tends to actually yield higher delivery efficiency. And this is because cells form a monolayer. You inject your solution onto the cells, and if they're really big, they just physically settle faster onto the monolayer. You know, for an in vivo strategy now, we're doing a lot of work with ML in vivo for injections, and generally we really want smaller particle sizes. So unfortunately, with what we've discovered with particle size in vitro is not going to necessarily translate to in vivo, and we have to do separate ML on all that data. And we're in the process of doing that. Now, with respect to lipids, the answer is absolutely yes. And we've extended a lot of this work in the company. I said, so like Nanite is a little more focused on polymers right now, whereas Liberate Bio is completely focused on using ML for LPNs and modulating chemistry. And they're doing a lot of work in vivo and have like a lot more capacity to rapidly crank through a lot of in vivo data for lipids and correlate chemistry, formulation, LPN size, all of the things to performance in vivo in a number of different animal species. So that is something, we're quite excited about too, to have sort of companies doing both non-viral strategies in the field.
Okay. We are almost it's almost towards the end of this session. And in the interest of time, maybe I will take this one last question because we had a couple of questions regarding this from the audience. So what will be your advice to young researchers who would like to venture into startups into the biomedicine field? And what are the key strengths of this technology over the existing technologies in manufacturing biologics? And what are the current challenges your startup is working on this technology?
Yeah. Okay. I will try to remember all these. So I'll maybe have
I
you,
can repeat, please.
Yeah. As I answer them, I might ask for your help to remember all of them in order. So, so my advice is, be resilient and flexible. It's been very educational and interesting to do this. And what I've really enjoyed, I have a whole team of scientists in my research lab, but I have, 2 other teams at companies. And right now I'm more involved in Liberate. I launched Nanite a couple of years ago and sort of, let them fly from my nest and off. And now I'm more involved in Liberate because that's a more recent venture. And, I've been involved in mentoring a lot of the junior scientists there. So that's been very exciting. I enjoy it a lot. It's different than academic research because they all have PhDs. I think from a standpoint that, they're young companies and the data they're generating every day is very key to the longevity of the entity and fundraising. And I think from a challenge perspective, currently right now, the biotech market is a little tricky and tough to raise major venture capital funds. So it's an expense. This particular field is expensive, and both my companies are in Boston, and that is an expensive area to have a company because the rent is high. So, you have to start out small, and it's okay to be small and use your kind of funding wisely as you go, because you can spend a lot of money very fast. So be wise, I would say, in spending some of your key seed money to try to get that key data. But you never know. And, look for opportunities, meet people, network, try to have that key difference, of technology and really what the key ultimate long-term goal is. There's a lot of ventures capitalist firms that like to fund kind of really pie in the sky, wild ideas, where some like to fund kind of more safer ideas. So when you're looking at funding opportunities, make sure you're sort of going, you read up on the history of funding and kind of where your VC sits if you're going to go into fundraising. And then if you could repeat, if I haven't answered one of them, maybe repeat the last.
No, I think you have covered everything. Yeah.
Oh,
And yeah, okay,
great.
this last part, maybe what are the current challenges for your startup working on this technology?
Yeah, I think right now we have a wonderful workflow and we have a lot of very exciting data. I think the challenges right now are basically in just raising funds. I mean, this is I'm preaching to the choir of like junior small companies, is just making sure we can kind of get enough funds to get that critical data to potentially go into phase 1 clinical trial. And I think we're, like, for example, we're right at that point trying to convince people to make, major investments. Right now, the market is a little bit tough in biotech, and we've had a depression, like financial sort of, during COVID, people were a little more willing to invest in risky ideas, where now I think the market has kind of fallen back, where they like to invest more in safe ideas. And so that's been a little challenging right now, but hopefully we have a lot of great data. I'm very excited about this company and their future possibilities. And hopefully we can find some other, we have a major investor right now. Hopefully we can continue to work with other investors as well to keep our entity going.
Okay. Thank you. With that, maybe we'll close the Q&A session here. Thank you, Professor Reineke, for answering all our questions so wonderfully. And we have a few more questions, but in the interest of time, we will skip that for now. And before ending the session of ACS Science Talks, please, I invite you to share with us any concluding remarks or any thoughts that you have with us.
All right. Well, in conclusion, I'm just so excited to be here. I want to thank ACS for their time. And kind of overall, I think from an advice perspective is just, things can get hard, things can seem easy. Sometimes you look at, for example, me, who's 25 years into my professional career, and you're like, oh my goodness, I could never do that. I was a student. I was a struggling student. I came from a meager background. I'm a first-generation college student, but it was really the science that drove me. I was so excited about the field, and I just really kept following my dreams. It was not easy. And, still things are hard. Of course, I've got to generate money. And, along your path.
Thank you, Professor Reineke. Very, very valuable words. And I'm sure your journey has inspired everyone present here today. And with that,
thank you so much.
Yes, thank you. And with that, I will close this session and hand over to you, Asha.
Thank you so much, everybody. It was a very inspiring talk to start off this week where we have International Women's Day, So thank you so much for inspiring us. And I'm sure the participants are showering their love and appreciation in form of emoticons. Thank you so much, participants. Yeah. And
Thank
with that,
you.
we'll close today's session. Thank you so much, everybody. Stay safe. Have a good day or a good evening, according to where you are in the world. Thank you so much, everybody.